Naked egg experiment. This is part of Lab 8 for Bio 1210. Making observations. You first want to carefully study the chicken egg. If you have a chicken egg, it would be a good idea to bring one out and study it, answering questions like, can you see through the shell? What is the composition of the shell? What makes up the egg? Research these questions using primary literature only and make your observations. In this activity, you will be conducting an experiment on a raw egg to see how different concentrations of salt solutions affect the egg. Vinegar will be used to dissolve the shell. Let's make our observations. The question you will be addressing today is, what effect do various salt solutions, 0%, 1%, 5%, and 10%, have on the chicken egg cell? Part of your observations is you should record what the anatomy of the egg and how are we going to assess the effect of various salt concentrations on the egg. The very first step would be to remove the shell so we could expose the outer membrane, the outer shell membrane, and see the effect of osmosis on the chicken egg. Begin with your hypothesis and prediction. Hypothesis. Write down your hypothesis before viewing or performing the experiment or continuing with this video. Immediately after the hypothesis, you'll write a prediction. The hypothesis could be either written in the null hypothesis or the alternative hypothesis. Remember, the hypothesis is an answer, an educated answer to your question. Refer back to your question. We're looking at what effect do the various salt concentrations have on the egg. Therefore, you could write your null hypothesis, there is no effect. The prediction would be what would happen if your hypothesis is correct. Again, if it's the null hypothesis, there is no effect. If it's the alternative hypothesis, there is an effect. The prediction would follow up, what is that effect if you're writing the alternative hypothesis? Activity 2, naked egg experiment. The materials you need for this experiment include 4 raw eggs, mason jars or any container that's see-through can be sealed with a lid, white vinegar, enough to cover all 4 eggs in a jar, salt, 100 grams total, water, about 4 liters of water, and paper towels. You can see the supplies right here in this picture in the bottom right-hand corner. Removing the shell. You'll need 24 hours to remove the shell. You'll pour vinegar in the see-through glass mason jar. You're going to add the vinegar to fully cover the egg. And let's see what happens. Looking at the equation for the reaction between the vinegar and the eggshell, what do you think are the bubbles forming around the egg? Let the egg sit in the vinegar for 24 hours. Rinse the egg shell completely off using tap water.
Naked egg.
And you can see that you have created your naked egg. You can see that the eggshell has been removed, is being removed, and you can see right through the egg, and the yellow is the yolk of the egg. It's soft and squishy. The white part of the egg, the albumin, and the egg yolk is still inside. We're pressing on the outer shell, the outer membrane. Preparing the salt solutions for submersion of your eggs. Remember the question, What effect do the various salt concentrations have on the egg? To view how to prepare the salt concentrations, there's a link in your lab to measuring and creating your salt concentrations, your salt solutions. Here I have listed briefly the procedures. Summary of the procedures. You measure out 100 grams of salt. 100 grams of salt will be added to your first container with 1 liter of water. This is measuring out 500 mLs. So 500 plus 500, 2 of these would equal 1 liter. You'll mix and dissolve the 100 grams of salt with the 1 liter of water to create your 10% solution. In the frame to the right, you have your 10% solution. So you mix well, you could warm it up to dissolve it completely. You'll grab 500 milliliters or half a liter of your 10% solution, place it in a container labeled 5% solution, add another 500 ml of water to create your 5% salt solution. Once you mix that very well, you're going to take 200 ml of your 5% solution into your 1% jar and add 800 milliliters of water to make your 1% salt solution. You'll have your jars ready for your experiment. The fourth jar that you need to create is 0% salt solution. 0% would mean there is no salt, it's just water, deionized water. Conducting the inquiry. You'll label your jars accordingly and then place each of your naked eggs into the jars. At this time, you could measure the eggs' weight in grams so that after the experiment, you could see if there's a difference in the weight. You're going to insert each egg into its appropriate jar and wait 24 hours to see the results of the effect of salt concentrations on the eggs. There goes the first egg in the 0% salt concentration, so that deionized water. Second egg in the 5%. And our last egg, naked egg, bouncy, soft, into the 10% salt concentration jar. Let the eggs sit for 24 hours and then record the results. This is the results. So visually, the results are here for 0%, 1%, 5%, and 10%. Here's a zoomed in picture of just the 0% egg and the 10% egg side by side. The table to the left has the salt concentration in the first column, the initial weight of each of the eggs in the middle column, and the final weight of the eggs in the third column, all measured in grams. So now you get your results and you make your interpretation. So you could write a discussion and conclusion based on this experiment. Your discussion should include explanation of the results, as seen here. Reference to previous research. Remember that you did research prior to starting this experiment. Do your results agree with previous research? If so, what did they add to it? Hypothesis. Remember, you had to write your hypothesis before viewing the experiment. Discuss whether the results met your expectations or supported your hypotheses. And explain any unexpected results and evaluate their significance. You'll write a formal discussion conclusion, only the discussion part, for this experiment in a post-lab 8 assignment. Write discussion conclusion. You're not writing an introduction, you're not writing results and showing the results that's given to you. You're only writing discussion conclusion for your post-lab 8 assignment. Again, they are based off the results that you need to interpret. So this is the naked egg experiment. Don't forget to write your post-lab 8 and turn it in on time. Thank you.